All right, then let's go ahead and get started drawing out mitosis and then comparing that to the process of meiosis. So starting with mitosis. Before we enter mitosis, the first part of our cell cycle, remember, begins in eukaryotic cells, that is. Keep in mind that for both of these processes, because we're talking about mitosis and meiosis, that implies we are talking about eukaryotic cells, okay? So, first process, or first part of the cell cycle for our eukaryotic cells is interphase, which, as you may remember, is divided into 3 subphases. We have our G1 phase, our S phase, and our G2 phase. So for the purposes of this drawing, instead of drawing out 46 chromosomes in each cell, we're going to let 4 drawings of a chromosome represent 46. Sound good? Okay, and I'll do my best to try to color code as well. So here in G1, we have our chromatin. So here are 4 chromatins. All right. And now during G1, the cell is existing and growing. And so transitioning from G1 to G2, we have our S phase, which, remember, stands for synthesis. And synthesis means DNA replication. So here we go. This is G2 now after S phase has occurred. So we have our DNA replication phase. So our chromatin now are going to condense into chromosomes as we enter mitosis. So this little dotted line here represents our transition from interphase to entering mitosis. Now remember, even though there are a lot of circles in this mitosis phase that you see here, over here in interphase is actually where the cell spends the majority of its time while existing. So let's break down mitosis now. Let's draw it out. First things first, during mitosis, in prophase of mitosis, we still have this nuclear envelope, right? And here we have our chromatin have now condensed into chromosomes. So I'm going to represent chromosomes by just these lines. And then that little dot in the middle, those are the centromeres. Okay, so there's a chromosome, and there's a centromere, right? So each one of those lines represents a sister chromatid that are bound together by a centromere, and now these are our chromosomes. So now during prophase, we have the nuclear envelope start to degrade, so we can erase part of our nuclear envelope. It's starting to break down. Our chromatin have condensed into chromosomes, as you see here. And we have spindle fibers starting to form. All right, so that concludes prophase. Now, moving on to prometaphase. Here we have the spindle fibers attaching themselves to our chromosomes. Here we go. So our spindle fibers are attaching to our chromosomes. They're not really pulling them in any particular direction just yet, but they have attached themselves to our chromosomes. And so that is the one distinguishing factor usually between prophase and prometaphase. Now, when we talk about meiosis, we're not really going to touch on the prometaphase because in actuality, when you're observing these processes under a microscope, you really cannot clearly distinguish prophase from prometaphase. Usually we just talk about prophase and then move on to metaphase. So here in metaphase, that nuclear envelope fully gone, right? It is fully dissolved. And this is the phase where our chromosomes are pulled and line up along the center of our cell, along a region of the cell that we call the metaphase plate. Now the metaphase plate, it's not a literal plate, right? It's not a physical structure, it's just an area within the cell that we call the metaphase plate because it is where the chromosomes line up during metaphase. Makes sense, right? And this is at the center of the cell, all right? So along the midline of the cell. Now moving on to anaphase. Here we have our sister chromatids being pulled apart to opposite ends of the cell. So we started out with our chromosome looking like this, right? And what I'm about to draw is chromosomes being, the sister chromatids within the chromosomes being separated. I'm drawing it like this, all right? So the way I want you to imagine it, imagine if somebody is grabbing you by your midline and then pulling you away from something, right? That would cause you to bend, right? And so that's what we see when we tug on those sister chromatids and pull them apart. They bend like that. And so that's what that illustration means, all right? So we're separating sister chromatids within the chromosome. All right, and now that we have wrapped up anaphase, it's time for, you got it, telophase. Telophase, telophase, however you prefer to pronounce it, all right? So during telophase, the key components here are, number one, our chromosomes are now at the poles of the cell, right? They've been pulled to the poles of the cell. So poles meaning ends of the cell, all right? And we have our nuclear envelope reforming. Then we also have the formation of either what we call a cleavage furrow. That's what we see in animal cells. So this little bit of cleavage here, this pinching in we see, that's what we call a cleavage furrow. So we either see that forming in telophase, or if we're looking at plant cells, here we have our little nuclear envelope starting to reform there. During telophase for a plant cell, we would have something more rigid, like this starting to form, and this is what we would call a cell plate. An important note to make here, though, is that telophase is actually the last phase of mitosis. So remember, mitosis is defined as the division of the nucleus, and so telophase involves the division and reformation of our nucleus and our nuclear envelopes. So once those nuclear envelopes are fully reformed, meaning we now have 2 nuclei, that means mitosis is fully done, all right? But cell division is not done yet, okay? The last part of cell division, not technically mitosis, but cell division, is cytokinesis. And cytokinesis is defined as the actual division of the cell, specifically the division of the cytoplasm. So here we have our nuclear envelopes, we have our chromosomes. Boop. Ooh, that's a messy chromosome. You get the idea. Got our chromosomes in each cell, and we have this pinching in here, this division of our cytoplasm between the new forming cells. And so once cytokinesis is done, then we have our 2 daughter cells produced from the process of cell division, mitosis and cytokinesis. Sound good? And so once this whole process is done, this whole process of cell division, those cells are now back in interphase. And so in theory, since they're in interphase now, they're eventually going to be ready to once again go back through the process that they just entered, right? They're gonna eventually go back into replicating their DNA during S phase of their cell cycle, then re-enter that G2 phase, and then go back into mitosis. Sound good? And so just to round things out, here, because we're back in interphase, our genetic material is back to being noodly in nature, so it's back to being chromatin. And if we want to compare these two produced cells to our original or parent cell, scrolling back this way, we see that they are the same. The only difference is that I forgot to make sure that these guys have their nuclear envelope. There you go. Yeah, but this is where we started with our chromatin, 4 representing 46. We had our 2 blues and our 2 oranges in this example. And if we go over here, 4 representing 46, each one of these cells has 4, meaning 46. And each one has 2 blues and 2 oranges. So perfect duplicates of the original cell and perfect duplicates of each other. Sound good? Okay. Well, with that, we are done with mitosis. So let's go ahead now and circle back to going over meiosis. So let's go ahead now and circle back to going over meiosis. So starting again with interphase. So we're in interphase where we have our G1 phase, our S phase, and our G2 phase, right? We have our nuclear envelopes during interphase are still there. And once again, we're going to let 4 chromosomes that I am drawing represent 46 chromosomes, right? So if we only see 2 chromosomes in a cell, that means that there's only 23, because 23 is half of 46, 2 is half of 4. Sound good? So that would mean that in this example, if you see 46 or 4 chromosomes, that means that we're looking at a diploid cell. Whereas if we have a drawing with only 2 chromosomes, that means we are looking at a haploid cell. Okay? So just keep this in the back of your mind as we draw our meiosis diagrams together. Sound good? All right, so starting out similarly to how we did before, we have our 4 noodly bits of DNA representing 46, right? So those 4 noodly bits, that's our chromatin, right? It's not condensed yet. But then after we go through S phase of interphase, the DNA is replicated. So once we're in G2 phase, that means we suddenly have pairs, right? There we go. And now we are ready to enter meiosis 1. All right? So starting with prophase 1, just like before, we have that nuclear envelope starting to break down. And now I'm going to zoom in on here so that way we can get a nice close look at what's happening during prophase 1. All right? So we have that nuclear envelope breaking down like we saw in our previous iteration of prophase. Also similar to our previous iteration of prophase, our chromatin have condensed into chromosomes. So here, once again, I'm representing each sister chromatid with a line, and they are connected to each other via that centromere. And we also have, like before, our spindle fibers starting to form. Now, this is where meiosis becomes truly unique, okay? Let's pretend that these 2 chromosomes here code for, let's say, hair color, just as an example. And we can say that these 2 chromosomes here, let's say that those code for eye color. So the 2 bottom ones are our eye color chromosomes, and our 2 top guys represent our hair color chromosome. All right, so something that happens during prophase 1 that's unique to meiosis, right, is we have something called synapsis. Synapsis refers to when our homologous chromosomes, meaning chromosomes that code for similar traits, pair up. So these guys are going to pair together. So I'm going to go ahead and erase and redraw that. All right, so we have our chromosomes that code for hair color pairing up and the chromosomes that code for eye color pairing up. And we can imagine that maybe the ones that are orange are the chromosomes that were inherited from the biological father, and that the blue ones could be the ones that were inherited from the biological mother, right? So there's one chromosome that codes for hair color from the biological father, one chromosome from the biological mother that codes for hair color. They're similar traits, and so they have synapsed together, okay? The other thing that can happen during prophase 1 that's really important for genetic diversity is crossing over. And a lot of textbooks in different places will also call this recombination. Crossing over is when homologous chromosomes twist around each other and they actually exchange genetic information with one another. And so the way that we would represent this in a drawing is if you can imagine these segments are crossing over and exchanging a bit of genetic information, that would mean that now this chromosome over here would have some of the genetic information from that blue chromosome, and this guy would have some genetic information from the orange one because they exchanged. Make sense? And so this is really important. Recombination / crossing over, really important for genetic diversity, because now already the cells that are produced from this process have no chance of being the same as the original cell, right? Already the chromosomes are different. And so genetic diversity is going up just from this first step of meiosis, all right? So that's prophase 1. Now let's move on to metaphase. I'm just going to abbreviate it, metaphase 1. So during metaphase 1, kind of like what we've seen before, our spindle fibers are grabbing chromosomes, but the key difference here is that what's being grabbed by the spindle fibers is now homologous pairs of chromosomes as opposed to individual chromosomes. So here we go, that guy has a little bit of orange, this guy has a little bit of blue. So now homologous pairs of chromosomes are what's lining up along the metaphase plate as opposed to individual chromosomes. So something that is special and important to keep in mind for metaphase 1, I'm going to go ahead and erase this just to give us a little bit more room. So something that's important that occurs during metaphase 1 of meiosis is what we call independent assortment. And so independent assortment is basically the term that we use to describe the phenomenon that the side of the metaphase plate, that each half of that homologous pair, meaning, you know, the one maternal, one paternal chromosome. So basically the side on which the metaphase plate, that each half of the homologous pair lines up, is completely random, right? So it's completely random. This chromosome could just as easily have been on this side of the metaphase plate, and this one could have just as easily been on that side of the metaphase plate. All right, they will line up together on that plate, but the side on which they line up, meaning the predeterminant for how they will be split up in anaphase, is completely random. They assort independently. Does that make sense? So that's independent assortment, and this is another key contributor to genetic diversity. So I'm going to go ahead and highlight that as well, because we don't know in which way the chromosomes will be split into their daughter cells by the end of this process. It's random, and that randomization increases genetic diversity. Make sense? Okay. So therefore, during anaphase 1, when homologous pairs are pulled apart from one another, meaning full chromosomes are being pulled as opposed to just sister chromatids, there will be a random mix of maternal and paternal chromosomes on each resulting daughter cell, and that promotes genetic diversity. So during anaphase 1, those paired chromosomes separate from each other, pulled by the spindle fibers to opposite poles of the cell. All right? Homologous pairs of chromosomes are what are separated, which is different than what we saw in mitosis, right? Because mitosis has sister chromatids separated. So moving on to now telophase 1. All right? So in telophase 1, we have our nuclear envelope starting to reform. We have our chromosomes at the poles of the cell. And so if this helps, I'm going to go ahead and say that this right here is what corresponds to that part of the cell. I realize I kind of turned my cell on you. So there you go. So we have our orange guy with the blue that had undergone recombination earlier over there, and then we have our blue guy with the orange guy that had a little bit of recombination over here. Okay, so we have our nuclear envelope reforming and our chromosomes pulled to the ends of the cell. All right, and now during our division of cytoplasm, right, our cytokinesis, I'm just going to abbreviate that cytokinesis, our division of cytoplasm, we have that pinching in, right? So it takes place at the end of our telophase 1 phase, and then we have our formation of that nuclear envelope finishing up. We have our fully formed chromosomes, and we have that pinching in, that cytoplasmic division producing our 2 daughter cells. Yeah? All right, now something that's really important to see here now though, all right, so now the cells are in interphase again. They're done with meiosis 1. They still have to go through meiosis 2 though. All right, so here we have our cells in interphase, but how many chromosomes are in each cell in our drawing, right? How many do we have? We only have 2 in each cell, right? So that would mean that already there is a significant difference from these initial daughter cells, not our end result, just the mid result. Already these are significantly different from where we started, right? Here we started with 4, and now we're down to 2 in each. So that means, scrolling back to our square from the beginning, we have gone from diploid cells to haploid cells. Make sense? Okay, so now let's go ahead and enter into meiosis 2. All right, so we can track each of these cells like so. So once again, we are back in prophase, but now we're in prophase 2. All right, so prophase 2 might look very familiar. All right, after the short interphase following meiosis 1 occurs, but there's no S phase during this interphase, and that's really important because no S phase means that we are staying haploid. Make sense? Okay, so after that brief interphase with no S phase, we enter into meiosis 2, which follows actually the same steps as mitosis, with the key difference being that instead of our cells being diploid, they are now all haploid. But otherwise, it's very similar. So prophase 2, we have that degradation of the nuclear envelope. We have our formation of spindle fibers. And we have our chromosomes condensing, or chromatin, I should say. Our chromatin is condensing into chromosomes. And now thinking through this, right, do you think we would have synapsis taking place during this phase like we did during meiosis 1, prophase 1? No, right? Because these are different chromosomes. These are not homologous chromosomes, so they cannot pair up and undergo synapsis. Does that make sense? If you want to go back and track it just to follow along with what I'm saying, here, these are the homologous chromosomes, right? So the homologous chromosomes were separated during anaphase. So if we're tracking it, these two chromosomes here were our hair color chromosomes, and then these two down here is what we represented as eye color, right? So now if we look at how the cell pulled apart those chromosomes, we have one eye color chromosome here, and this guy is a hair color chromosome, right? It's this guy right here. All right, so these guys are different traits. These code for different things. They would not synapse together, right? These are different. So no synapses, no crossing over here. Just normal prophase. Sound good? All right, so chromatin condenses into chromosomes. Each chromosome is made up of two sister chromatids joined at the centromere. Spindle fibers are forming, nuclear envelope is breaking down, and the cells are haploid. Now we're going to go ahead and move into metaphase 2. All right? Metaphase 2, our chromosomes are lining up along the metaphase plate. So thinking through this, do we think that chromosomes are going to be pulled apart or sister chromatids are going to be pulled apart during anaphase? What do we think? Chromatids, right? We're going to be splitting these guys in half during anaphase. And so that would be pulling apart sister chromatids because there aren't pairs of chromosomes. Make sense? So during our anaphase 2, those sister chromatids are what get pulled apart, just like what we see in mitosis. So our sister chromatids are what get pulled apart during anaphase 2. All right. Now we have telophase 2. Which remember, you got it. That's when we have that nuclear envelope starting to reform. Our chromosomes are at the end of the cell. So just for clarification, we're going to say this is that pole there. This is that pole there. I drew it sideways again and I apologize. Here we go. There we go. Just for clarification. All right. So here we have our telophase 2, where chromosomes are at the poles of the cell. There are no spindle fibers. Those spindle fibers have been disassembled. The nuclear envelope is reforming. Here we go, reforming. And then the cell concurrently enters cytokinesis, which is the division of our cytoplasm. Round off that cytokinesis process, forming our final product of meiosis, which is our 4 daughter cells, right? And if we want to evaluate these, I'm just gonna draw in our nuclear envelopes, and our chromosomes decondense back into chromatin because these guys are now in interphase again. Here we go. Granted, these guys are not going to go through meiosis again because you can't do that again. Otherwise, you would be at 1/4 the number of chromosomes you need, and that's not what we want. All right, so evaluating these 4 new haploid cells, right? Well, first things first, are they actually haploid, right? Are these 4 cells diploid or haploid? Well, let's compare, right? Zooming out all the way back, right? They're haploid. They have 2 chromatin, or 2 pieces of genetic material representing 23 in each cell. So they are haploid in nature. And now let's compare them to where they started, right? They have half the number as when they started. They started diploid, ended haploid. Now comparing each one to each other, if we compared these 4 cells to where they started, are they genetically identical to where they started? No, they're entirely different from that starting place, right? How about to each other? If we compare each one to each other, right? Comparing this one and this one, comparing this one and this one, comparing this one to this one, comparing that one to that one, right? If we compare these to each other, are they genetically identical? No, not at all, right? And that's because, and big thanks to the fact that we have crossing over or recombination occurring during prophase 1. We have independent assortment occurring during metaphase 1. And when they go into interphase between the 2 rounds of meiosis, no S phase occurs. All right? So these all contribute to the haploid nature of these cells and genetic diversity, which is super important. Sound good? All right, so now what you're going to do on your own as one of your activities is you are going to compare this process of mitosis to this process of meiosis. Sound good? And I tried to line it up for you in a way that I felt might be conducive to help you with this comparison. Sound good? All right, and with that, I hope you found this helpful and best of luck.